Okay, so let's start. Good afternoon, everyone, or good morning for those of you that are joining from a different region. Welcome to the second webinar of the Virtuous Massive Open Online course, Promoting Ocean Literacy and Project-Based Learning by Monitoring Biodiversity. You may know me from the last week webinar. I am Tekla from Sumon, one of the partners of Virtuous Project, and I'm pleased to introduce you today's presenter, Dr. Sally Deng. Hi, Sally. Hello.
Hi.
She is a marine biologist and she currently works at Guilmar Helmholtz Center for Ocean Research based in Kiel, Germany, that is a partner in Virtuous Project. Today, she will give us an overview of what is biofouling and how can students learn about biodiversity through the observation of Virtuous discs. Before handing it over to Sally, I would like to remind you that your microphones are by default on mute and you are not able to start your video. If you have any questions regarding today's presentation, please post them on Canvas platform using the forum of Module 2. Sally, in the next few days, will clarify your doubts and answer your questions. Well, now I'll leave the floor to you, Sally, and see you later.
Okay. Hi, good evening, everybody. So I'll just remove my picture here so you will just see the presentation on your screens. We have posted in Canvas a PDF version of the presentation, but I have made last-minute changes, so that is not the final version yet. The final version will be posted after the webinar, probably tonight or tomorrow, and then Tekla will be recording the presentation anyway, so you can just you can look at the presentation at your leisure after the webinar. If there are some things you want to clarify or some things which are not clear during the presentation, you can go back to the recorded version. Okay, so this presentation is about biodiversity and fouling, and this is a follow-up of the presentation of Michael and Susan about the Virtue disc, and I'm going to talk about what happens in the Virtue discs when you put them in the water, and also how are you going to work with the discs after you have retrieved them from the water. So the topics I will be talking about today are outlined here. So I will be talking about biofouling, a little bit about it, and then what is ecological succession, and how are you going to calculate biodiversity. So some pointers on how you can do that with your students, how to calculate the biodiversity on the discs, and then some drivers are biodiversity. So what are the parameters or what processes can change the biodiversity in an ecosystem? After that, we are going to have, that's the first part, we're going to have a 3-minute break afterwards. And after that, and you can have a drink of water or get some sandwiches from the kitchen or pizza, or order a pizza and eat during the second part of the presentation, where I will be talking more on the theoretical part about biodiversity and ecosystem functions, and what are the applications of fouling discs in research. And then I will give a short summary of the entire webinar, or a short summary of what we have talked about, or what I have talked about during this webinar. I have here a sign or a symbol. This is what you will find in the PDF file of the webinar, because some of the, or most of the animations which I have in this presentation will not be placed in the PDF. And these animations are available as video bits or short videos, which are also posted in Canvas. So if you want to look at these animations, you can look at them in Canvas. You can also use them in class. If you want to use them, you can download them from Canvas and use them for your class. Okay. So let's start now. So the first part is, what is biofouling? We have biofouling is more where you can see here both accumulation of undesired things, or undesired deposition on any surface or on surfaces. Fouling can be physical things like organic substances like oils, fats, and proteins, inorganic substances like calcium carbonate and salts, and some abiotic particles like clay and silt. And then you also have biofouling by biological materials and surfaces, which will be, of course, so biofouling would be plants, animals, and protozoa. So biofoulers can be big or small. So they are microfoulers, which are the microfoulers which are microscopic, and these are bacteria, fungi, or microalgae. And you have the larger organisms, the macrofoulers, which may be hard-bodied or soft-bodied animals, mostly invertebrates. And on the discs, there is a combination of fouling and biofouling. And here is an example, what we find mostly in a lot of the discs that we deploy here in our area. There is, here is the biofouler. Okay, that is a tube worm, and here you have the silt, that's the physical or the abiotic material, which is actually needed by this tube worm to build its tube here. Okay. So in the formation of the biofilm, start here, you see here that the bit for biofilms and virtue discs, you can look at this, the animation of this in the Canvas platform. Okay, so now we have the disc, the virtue disc, deployed underwater, and this discussion is just a model. So this is just a model of how biofilms are formed, and this is what happens regardless of the type of substrate. May it be plastic or rocks or stone, or even if it's just a leaf from a macroalgae, what happens during the early stages of fouling will be the same for all kinds of substrates. There are 4 stages in the formation of a biofilm, and the duration for each stage depends on the location and time of the year. So the time frame that I will be discussing here is just, again, a model. It is different in every location and the time of the year. So this shows the earliest stage of the fouling. Okay, within seconds, when the disc is placed underwater, there will be the process of adsorption and adhesion. Okay, there is first the biochemical conditioning of the substrate. Biochemical in terms of there will be first adsorption of chemical compounds. Okay, or adhesion, adhesion of chemical compounds, which are macromolecules like glycoproteins, polysaccharides, and proteoglucans, which they just adsorb to the surface. The process of adsorption here is purely physical, and it is spontaneous. So it is just what is present in the environment, what is present in the water, what will determine what kind of chemical compounds will be adsorbing to the surface. After a few hours, dynamic equilibrium is reached. The surface, it will already change, of these chemical compounds will already change this characteristic of the surface of the disc, turning it from being hydrophobic, if it's hydrophobic originally, to become hydrophilic, which is water loving. So it becomes more wettable. The next stage would be the settlement of bacteria. The settlement of bacteria is, again, also at the early stage also physical. physical forces like Brownian motion, electrostatic interactions, gravity, and van der Waals forces are the ones governing the adsorption of bacteria. Okay, there are two phases of bacterial colonization. First, you have adsorption, which is reversible because they are not yet fixed on the substrate, and there is adhesion, which is irreversible. Okay, so the process of adsorption is purely physical, and the process of adhesion, that is when the bacteria start really being attached to the surface. That is when biological processes start, like for instance, when the bacteria start producing microfilaments, which will attach them to the surface. Okay, so there is, see now? There is still a debate which comes first, the diatoms or the bacteria. So in most cases, there are, in a lot of studies, some say that the diatoms come first before the bacteria, and others, some say bacteria come first before the diatoms. Whichever comes first, both of these organisms, okay, help each other to settle under this because they produce something which is beneficial for the other. The bacteria, for instance, produce much, some of them probably may be fixing nitrogen, which is important for the microalgae, or the microalgae may be producing organic material, which the bacteria can use as substrate for their metabolism. So within hours and days, this conglomeration of bacteria and plankton, diatoms, these are not plankton, these are diatoms and microalgae. Okay, this conglomeration will grow and divide, and simultaneously producing all these biofilms, excretions, and mucus, which they produce during their metabolism. During this time, some will be dying in the process, and some will be multiplying in the process. And also, again, take note that this will not be the same bacteria all the time and not the same microalgae all the time. There will be recruitment or new bacteria, types of bacteria and types of macroalgae, which will be joining them in this surface. So after growth and division, the biofilm will mature. And then after some days, some of the bacteria will start to disperse. Okay, so we have here a time frame from seconds to days, but this is just a model. So in some cases that may take longer, in some cases it may be faster. So it depends on where the disk was deployed. It will be, the time will be, when that happens will be variable. Here is an example of a plate, okay, made of high density polyethylene, which was deployed for 4 months in the US, okay, from July to November, okay, and these are scanning electron photomicrographs of the plate. So that is the time 0. So in this case, after a week, we have here the diatoms. Okay, the first things to settle here are the diatoms, but there are also bacteria already here. Okay, and then after 2 weeks, the bacteria start settling in and the diatoms start dying out. These are the frustules, the leftover frustules of the diatoms. And then here, after 4 weeks, you have more bacteria than diatoms. And then 8 weeks, they start dying down. And 6 weeks, you have a conglomerate of different bacteria. And if you study and try to identify what these are, you have at the first week here, you have more diatoms than bacteria. And as the weeks go by, the number of diatoms decreases and the number of bacteria increases. Okay. The type of bacteria or the type of bacteria which grow on the substrate will depend on interactions which are going on between the early bacteria settlers on the surface. There are some studies even which have found out that the type of bacteria which will settle on a substrate depends on the type of substrate. For instance, there are new research now which use different kinds of plastics, and then some of these studies have found out that a different type of plastic will attract a different type of bacteria. Okay. But what is common to all of this is that the first settlers are these microorganisms, the diatoms and the bacteria. That is common to all. It doesn't matter what type of substrate, it's bacteria and diatoms and some protozoa. Okay, and then what matters or what is different is the type or what kind of bacteria and what kinds of diatoms are present as early settlers. Okay, so now we have the biofilm. Okay, and then the next thing that happens would be there will be the settling of the larger organisms, the multicellular organisms, which will be normally soft foulers if they are present in the water or if they are present in the location, like tunicates here, the anemones and hydroids, or if there are spores in the water, macroalgae or green algae spores, you have also macroalgae, and then this will be followed by the hard foulers like the barnacles, mussels, tube worms, and the bryozoans. So going back again to the processes which are happening during the early stages of fouling, you have first the physical processes like the adhesion and adsorption of the macromolecules, the chemical compounds, where the substrate will be biochemically conditioned. So this biochemical conditioning of the substrate, and then at the later stage, when the bacteria starts settling in, then the biological processes which govern the fouling of a disc will start getting the upper hand, whereas the physical processes will become less and less the determining factors, which will determine what and how the organisms will start settling on the discs. Okay. So what are the consequences of biofouling? There are economic, most of them are economic consequences, like we have ship hulls, which are here, you can see here a ship hull. It's almost unrecognizable because it's already full of fouling organisms. And if fouling organisms will grow on the ship's hull, and that in that big numbers, then they can reduce the ship's speed and may cause extra drag, increasing fuel consumption and engine stress, because the engines will start to work over time so that they can be moving faster. Another one is it helps in, or it will fasten. Okay, it will be biological corrosion becomes faster because of acid producing bacteria in the film. Acid producing, of course, because the bacteria are respiring and producing carbon dioxide. So it will be, or there are some types of bacteria which are also acid producers. So they will help, they will then also speed up biological corrosion. Another thing is we have already, we can have all experienced this probably when we go to the sea and then on the pier and go down the steps, and the steps will be covered with algae and they become slippery. So they create algae biofouling, can create slippery coatings. So it will be more dangerous and slippery. So there will be increased safety risks. And then another example for consequences, economic consequence of biofouling is that in aquaculture, for instance, if they use nets here, if for their fish cages, the nets will become clogged with biofoulers. Here you have some barnacles and mussels, okay? And then the nets will become very heavy, and then they could damage the nets. So they have to clean the nets several times, or they have to clean them more often, or they can even destroy the nets and they have to change the nets more often. Another one is blocking and clogging of cooling pipes. A lot of factories or even reactors, okay, use salt water for cooling, and if they suck in seawater into the reactors, to the cooling pipes, there will be organisms in the water, and they will start settling on the walls of these cooling pipes and then becoming, so here you can see that the diameter of the cooling pipe has already decreased, so that that becomes less efficient. Okay, and then so that this all cause enormous costs for cleaning and then eradicating the foulers. Another consequence of biofouling is the biological consequence. Okay, this is that the fouling of ship's hulls is an important vector for invasive non-indigenous species. So if you think about invasive species, we always think only of some of the species being carried in the ballast tanks of ships, and then when the ballast tanks will be emptied into the to another location, then the organisms in the water will be introduced into the new location. But that is not the only way of transporting non-indigenous species, but also those things which are already, those organisms, foulers, which are already on the hulls, they can be also transported to the other new areas, the new locations. So here are some examples of non-indigenous species which have become invasive in the new locations, where they were transported. Okay, so now we go to ecological succession. During the formation of the biofilm, we have seen already that there is an orderly and predictable order of events, a predictable change, okay, in how the organisms or how the things which adhere to the surface followed a certain sequence, okay, of appearance. So we had first the macromolecules, which don't count, of course, because they are just chemical, but then you have the bacteria or the diatoms, and then the bacteria themselves. Okay, there are early types of bacteria, which will be the pioneer bacteria species, okay, followed by different types of bacteria, as we have seen in one of the slides before. Okay, now after this microsuccession, so we can call them microsuccession because those are microorganisms which we find. So these are the diatoms, okay, which is also part of the microfoulers. Then we have the filamentous algae, and then these are followed by bryozoans, hydroids, barnacles, and tube worms, and the mollusks. So these are soft foulers, okay, and then followed by the hard foulers. So this is more or less the succession, the order of appearance of the organisms on the disc. But this is not always the case. It depends on what organism is available in the locality. Okay. So what will appear or what will settle on the disk will have to be present in the location where they are found, where in the location where you have, where the disks were deployed. So if there are mussels there, it is 100% sure that there will be mussels on the disks later on. And if there are no mussels there, there will be no source for the larval stages, which will later settle on the disk. Okay, so we have here, again, in the early succession, we have the bacteria and the diatoms, which will still form the biofilm. And this biofilm will be composed of polysaccharides and glycoproteins. Okay, and then we have the macroorganisms now, which will settle on the disk. Many of the organisms which will settle on the disk were once part of the zooplankton. They are larval forms of the adult stages that we find on the disk. So most of the things that we will find on the disk will just be the adult forms of these organisms, which we find in the zooplankton. These zooplankton are called the meroplankton because they are only plankton in the early, or at least once in one stage of their lives, and usually as larvae or in the young stages. So here are some of the larval forms, and these are some of the organisms, fouling organisms that you will find later in the disk, the bryozoans, the mussels and snails, and so on. Okay. If it is possible, if you have a zooplankton net in your school, or if you have a zooplankton net available, that will be, it would be nice if you could get zooplankton samples from the location where you are transporting or where you are deploying your disks, and it will be good to see if you can find some of these larval forms in the water. And if you can see these larval forms, then it would be nice to see if this will be the adult forms which you will find, if there is a correlation of the adult forms that you will find later on the disk with the larval forms that you find earlier in the water. Okay, so again, we have here, we have the intermediate stage, which will be the macroalgae, some tube worms, and some barnacles. And the later part, which will become the climax community, at least the climax community here in Kiel in Germany, or in the Baltic, our climax community is usually composed of mussels. Okay. So if we are going to characterize the different stages of succession, we have early succession, the intermediate stage, and the climax community. In terms of species richness, in the early succession, there are low number, the number of species will just be limited. Okay. And in the intermediate stage, you have a high species richness, and in the climax community, it will become low again. Why is it high in the intermediate stage? Because in the early succession, in the intermediate stage, you will have some organisms which were still in the early succession. So there is an overlap here in this part where you will have some species still from the early succession stage. And the same thing here, there will be an overlap wherein you will already have some organisms from the climax community growing here in the intermediate stage. So there will be, and in addition to that, there will be some other species which will be trying to settle on the disk, and then there will be a kind of sorting out which of these organisms will be the most adaptable, which organism is the strongest, and which organism or which species can in the end be the strongest among them all, and that will be the one which will be able to persist to form the climax community. Okay. Now, the number of individual species or number of individuals per species in the early succession is high because of course there are less number of species, so the species can produce more individuals. And here, the one thing that we should consider in the disk, the most important and the most limiting resource on the disk is space. Okay? The disk that we have is very limited, so that it can only support a limited number of species and a limited number of individuals. So that if there are a lot of species in the intermediate stage, the species cannot spread all over the place because there is a limited space, so that the number of individuals per species during the intermediate stage is low. And then in the climax community, it depends what the size is of the organism which will persist in the end. So it can be high if they are small, if the size is small, or they can be low if the organisms, the size of the species, or the size of the individuals of the species is high, if they are big. Okay. Again, this is easy to understand because species interactions would be simple if there are only several species, a low number of species. And here we have a complex because it is here in this stage where the organisms will be acting for each other, competing with each other, fighting with each other, trying to push one species away and another species working together with another species. So there is a complete complex species interaction because at this stage of succession, this is where the best species, the fittest species, will start to dominate over the other species to form the climax community, wherein, wherein it will just be simple if the species number here, species richness is low, okay? Then resilience, that is the how stable the community is in the early succession, is still high. Again, in intermediate stage, it is variable because it depends on what organisms are present there. There may be tolerant species or there may be not too tolerant species. And then again, here it is low because it is almost like a monoculture. So that the resilience of the climax community, if there will be a change in the climax community, it will be a big one, okay? Because the resilience is low. If there is a disturbance, it will be easy to remove these organisms because the organisms here will not be very tolerant. And in the trophic levels, okay, because they are of the low species richness, okay, the trophic levels here would be low and simple. More here is more complex, and again, low and simple in the climax community. So to summarize that table, in terms of species richness, the curve will look somewhat like this. The species richness is low in the early succession, it rises in the intermediate stage, and in the climax community it decreases again. And in terms of the number of individuals per species, it is the other way around. We have in the early succession, there is a high number of individuals per species, and decreases here because there are a lot of species here, so that there cannot be too many individuals representing one species. And then again, it rises here where if there is only if there are only a number or a smaller number of species in the climax community, then they can have higher number of individuals. Okay, in terms of interspecific and trophic level interactions, in the early succession, it is simple, and in the intermediate stage, it becomes more complex, and then it becomes simpler again in the climax community. Okay, so after several, depends on how long you will put the discs in the water, you will retrieve it. And depending on how long, you may get a disc which is like, which will look like this. This is, for instance, a disc which was deployed in Barcelona, I think, from one of our partner schools. And this is how the disc looks like. And the students, when they see that, what organisms are growing on the discs, they will be overwhelmed, especially if they see the live organisms. Okay. When they look at it and when they observe it under the stereo microscope, it will be hard to get them away, to pull them away from the stereo microscope, because they will be very fascinated with all the organisms moving there, because of the live organisms moving there, because it is not very common to see these organisms, especially the small ones, because it is easier actually to see living organisms if they are big. But if they are small, and if you observe them under the binoculars, under the stereo microscope, it is really fascinating. Okay, so that is where the fascination comes in if you work with a VirtuDisc project. So it will be really motivating for students. Okay, but the less motivating part and the more difficult part is when you put science now into the picture. Okay, if you ask them, how do you know if this disc, if the biodiversity in this disc is high or is it low? Okay. So when you say that you should start calculating biodiversity, the motivation probably will become less. So we have observed this several times already with some of our projects here in Germany. The students are always motivated looking at the organisms and observing them under the microscope and the binoculars. But when you say, okay, start calculating biodiversity, then the motivation drops. Well, anyway, they have to go through it. Okay, the first thing that they have to do, of course, is to identify the species or identify the organisms that they find on the disc. We have identification plates on the Virtu's website, and which can be used for identifying the organisms which found on the plates. But again, it will be a really big challenge for the students to identify all the organisms that they will find. And what we have done for the Virtus project to make it easier for them to calculate biodiversity is to create a drawing, a sort of a drawing of how a plate may look like, okay, and it would be easier with this drawing how to calculate biodiversity. Each color and form represents a species, and what they just have to do is count how many of this and how many of these are there. Because counting, for instance, these organisms here, like if you have the tube worms here, for instance, if you count the tube worms, it would be difficult without experience. The students are most of the time likely not too experienced in counting the organisms, but they have to. So if they want to calculate biodiversity, and then they can use these dry discs for practice. Sometimes also, if the discs are not as nice as this, are not as beautiful as this, it would be also difficult to find some really very nice discs to count. So practicing in the laboratory or in school can be done using this type of drawn disk. You can also ask your students to draw their own disks and exchange them with each other so that one group can draw or fabricate a drawing of a disk, and another group can start calculating the biodiversity from this disk. Okay, so they can start calculating biodiversity by counting two parameters, actually. The total number of individuals for all the species, which is the N, and the total number of individuals per species, which is the small n. So these are just the two parameters which are important to count, okay? I say just because it's just two, but it is not really that simple if you are going to count real organisms on the disk. You have to have, the students really have to have patience in counting them. Okay, so we have here the different indices of diversity, which we are usually used in ecology. And for high school students, these are easy to calculate. Have one is species richness, Simpson's, Shannon-Wiener, evenness, and the Jaccard index, which is a Jaccard index of dissimilarity or similarity, wherein we can compare two disks, the organisms or the species which are found in the disks. And these are the formula. Again, so we can see here again, formula, okay. So students will see a formula, oh, it is a formula, it's mathematics. Again, there will be, the motivation will drop again. But if they start doing this, actually, it's not very complicated, okay? So don't bother yourselves with this formula, because you can get this in one of the resources that we have made for you available on Canvas, as well as in our website. You can see this formula. Okay, so what do these diversity indices, what would these indices of diversity tell us? Okay, so here we have species richness, which is just a simple count of the number of species. Okay, plain enough. And then Simpson's index of diversity is the probability, okay, that two individuals you selected randomly from a sample belong to different species. I will illustrate this later, but so that it will be better, it is easier to picture. And so it is a measure of probability. Okay. So the closer D, the Simpson's index, is to 1, the higher the diversity. The Shannon-Wiener index is a measure of entropy. It is a measure of the uncertainty of the identity, okay, of a randomly selected species from a sample. Again, I'll show you how this is in a picture later on. And the higher the H, the higher the diversity. The higher the Shannon-Wiener index, the higher the diversity. And then from the Shannon-Wiener index, you can measure evenness, which is the measure of the relative abundances of the species. And the closer E is to 1, the more species are equally common. Diversity is higher. And the Jaccard index, as I have said earlier, compares the species composition of two samples. But here, the abundances are not considered. Okay. To illustrate this in a picture, I have here, let's say this is your ecosystem, and I have here each circle, okay, each color is a species, and each circle is an individual of the species. Over here, I calculate the different diversity indices of this. Species richness is 4, that is the red, yellow, green, and blue, so there are 4. And then for each of this color, for each species, there are 4 individuals, so 5 individuals. 1, 2, 3, 4, 5, 6, sorry. There are 6 individuals. Okay, I think I'm getting cross-eyed now. There are 6 individuals, and the Simpson's index is 0.78, and the Shannon-Wiener index is 1.38. This doesn't tell you much right now, okay? Simpson's index doesn't tell much. Is it high, is it low? And also the Shannon-Wiener index cannot tell you much, is it high or is it low, until you have another sample which you can compare it with. Okay. With this sample, you also have the same species richness. You still have the red, the green, the yellow, and the blue, okay? But the numbers are different. If you're going to calculate the biodiversity indices here, you have species richness is still 4. The Simpson's index becomes lower, 0.69. The Shannon-Wiener index is 1.24, and evenness is 0.89. Okay, so let's go back here to this sample. When I say here, Simpson's index is 0.78, okay, that means when I close my eyes, okay, I close my eyes and I get 2 circles or 2 balls from this ecosystem, 2 balls from this beaker or from this pail, okay? The probability that these 2 balls that I will get from this sample are different is 78%. Okay. Comparing it to this sample, if I close my eyes and get 2 samples from this pail, okay, what is the probability that these 2 balls or these 2 circles which I will be getting from this sample will be of different colors? The probability would be lower now. It will just be 69%. Because here, we have here, can see that the dominance, or there are more red individuals than compared to the other colors, okay? So it reduces the possibility or the probability that I will be getting 2 different colors from here is lower than from this sample where all the colors are represented in the same, with the same number. Also with the Shannon-Wiener index, okay, if I get a sample here, okay, and I don't see that, so then the uncertainty, I am more uncertain to know what I will get from this sample. Whoops, sorry, whoops. Okay, whereas if I am going to get a sample from this, okay, to get a species or get a ball from this sample, okay, I can probably say, well, I'm going to get a red ball because I would know that there are more red balls here. So that the uncertainty here is lower. Okay, here the uncertainty is higher, here the uncertainty is lower. The evenness here is 1 because each color has the same number. So there are, let me count again, 1, 2, 3, 4, 5, 6 reds, 6 greens, 6 blues, and 6 yellows. So the abundances, the relative abundances of all the species is the same. So that is why the evenness is 1. Whereas here it is lower, okay? Because red predominates. It is 3 times more than the other colors, okay? So that is why evenness here is reduced because the relative abundances is not the same for all, or the abundances for all the species is not the same, okay? So if evenness is 1, that means that has a high diversity, and if the evenness is low, the diversity is lower. Okay, your assignment later on is to calculate and compare the biodiversity of these 2 discs. So there's already on Canvas a prepared protocol sheet where it would be easier for you to calculate the biodiversity of these 2 discs. Okay, Now, sometimes it will be difficult to count the number of species and the number of individuals, the number of individuals per species. So that is really a challenge for many students, especially in school setting. If the time is limited, there will not be a time enough to count all the organisms on the disk. So what we have resorted to sometimes with our projects here with classes is to just estimate the percentage of the disk covered by a species. Here we have an example of barnacles, because that's what we get here. Unfortunately, in the Baltic here in Kiel, we don't get very nice discs like in Barcelona or in Spain, where they have a lot of nice exotic organisms. Here in Kiel, we just get barnacles and mussels. Well, anyway, we have to do the best out of it, of what we have. Okay, so we can estimate the percentage cover of a disc covered by a species. We also have a guide for prepared a guide for the visual estimation for percentage cover also on the Virtuous website. And here the students can more or less so estimate how much of the disc is covered by barnacles. So here are some guides. And then also we can see here also, well, at least we have also bryozoans here in Kiel. Okay, so we have here bryozoans and can also estimate how much of the disc is covered by bryozoans. So that is one shortcut. So that is an easier way to quantify what will be, or what the students will be finding on the discs. Okay. Before going to the drivers of biodiversity, if it will be very difficult, for one tip what I can give, or one advice what I can give to some teachers, is that if it will be very difficult to identify the species up to the species level, it is enough if they would be able to identify it up to the genus level. But also the biodiversity indices can also be used for up to the family level or the class level. Say, if you have a disc, and then you can say how many worms are there, how many polychaetes are there. Even if there are different species of polychaetes there, you can lump together the polychaetes and count them as one of the species. And then if there are mollusks there, then you can count the different species of mollusks as one. Okay, so that you will have not species diversity, but say family diversity or class diversity. So it will be easier for the students if they don't have to identify all the organisms or identify and count all the organisms on the plate. Okay. In Kiel, we don't have any problem about that because there isn't much to count. There are many species to count. Well, anyway, so the drivers of biodiversity on the disk, or not only on the disk, but generally, can be increased or decreased, but most of the time, when humans come into play, okay, there will be a decrease in biodiversity. Well, hopefully there are some environmentally aware people. Well, there are lots of environmentally available people now, so that human caused disturbances doesn't always lead to decreased biodiversity. Well, anyway, many of the human activities that we have, okay, cause decrease in biodiversity. So one of the human caused drivers of biodiversity are biological invasions, which we have discussed earlier, chemical inputs, so different types of pollutants and nutrients, okay, nutrients which can lead to eutrophication, for instance, if there will be too much nitrates and phosphates being put into the bodies of water, then there can be, that can lead to changes in the biodiversity. And then climate change, very important. Okay, it's very actual, it's very actual topic now, climate change. And carbon dioxide increase, which will lead to ocean acidification, and then exploitation or over-exploitation of the habitat, which will lead to habitat loss, degradation, fragmentation, and loss of species because of overfishing or too much hunting. Okay, so comparing the impacts of these different drivers of biodiversity on the marine and terrestrial ecosystem, both in the marine and terrestrial ecosystem, the most important driver of biodiversity, human-induced biodiversity, human-induced driver, is the use of land or the use of sea. Okay, land that is agriculture, and sea use that is more, so whatever we use the ocean for getting food, for as food source, or for getting other resources like energy and medicine, or also for recreation. So this is the most important anthropogenic disturbance, okay, which impacts terrestrial and marine ecosystems. And then in the marine ecosystem, climate change comes in second, and chemical inputs are also important. So land use, chemical inputs, and climate change are the most important drivers of biodiversity in terrestrial and marine ecosystems. Okay, so that was the first part of this webinar. We have a 3-minute break. The second part will be very theoretical, so some may want to get their pizza or eat their sandwiches, and they can come back in 3 minutes, or since this is also being recorded, actually they can also skip it, or but if you still want to hear my voice, well, we can you can join in the second part of the webinar. So I think Tekla will have
Okay.
something.
Thanks, Sally, for this first part of your presentation. So let you drink a glass of water and rest a bit before continuing. And in the meantime, I would like to remind you that you can use the forum of Module 2 on Canvas platform to post your question for Sally. And if you navigate to the end of Module 2, you will find the assignment that Sally prepared for you. So it's a multiple choice quiz to test your knowledge. And also, please remind to submit the assignment of Module 1 if you haven't submitted it yet. And at the end of the course, we will issue the certificate of completion of this massive open online course only to those that have submitted all the assignments. Well, so let's have a slice of pizza or just rest for 1 more minute, and let's wait until Sally is ready to start again with the second part of our presentation.
Oops. Where am I? Okay, so we have talked a lot about the biodiversity, about species richness, Simpsons, and the Shannon-Wiener indices of biodiversity. And these are actually really very good indicators of biodiversity. They are still being used now in ecology, but this, of course, and in research, but these, of course, have also their shortcomings. For species richness, for instance, the species richness alone is just an information about how many species are there. There is no information, it doesn't give us any information about abundances and the distribution of the organism. So just using species richness doesn't tell us too much, okay? And the Simpsons and Shannon-Wiener indices are not linear. I will explain that later. but also the Simpson-Shannon-Wiener indices does not give us any information about the structure of the community and the stability of the community. So what I'm talking about, for instance, for species richness, I have here two discs, okay? Both have a species richness of 3, okay? So here I have three kinds of species, and here I have 3, but you can see that these are very different. So you cannot say that both have the same biodiversity, because it doesn't tell us too much, except that there are 3 species found on the discs. Now going back to our example for biodiversity, in this sample 1, where we have a species richness of 4, and this different, where there is also, oh, I just see that there is wrong spelling here, evenness, okay, of 1, okay? So if I increase the number of species twofold, so of this, in this sample, so I have now 8 samples, of course, the Simpson's biodiversity index will increase also, as well as the Shannon-Wiener. The evenness will still be the same, because each new species will have the same number as the original. Okay, so they are still evenly distributed, or they will have the same abundances. Increasing again the number of species 4-fold compared to the first sample. Okay, I have here 16 samples now, 16 species now. The Simpsons increases, the Shannon-Wiener also increases. Okay, so that means the probability of getting, if I get 2 samples here, the probability of getting 2 different species is higher, according to Simpsons. And the uncertainty that I will get here, what I will get here, will be what I will be predicting, is also low. So the uncertainty is high. Okay. So, and then I have another hypothetical sample here with 8 times more than the original. So we can see here that the Simpsons and the Shannon-Wiener index are increasing. Okay. So what is the problem here? The problem is, if we are going to plot the Shannon-Wiener index and the Simpson's biodiversity index, okay, in relation to species richness, okay, we have here the samples here. The line is not linear. So the increase in diversity is not linear. So what? Okay, so if it's not linear, what's wrong with that? Okay, so going back here. So if we have an ecosystem, for instance, which originally had 8 species, and a disturbance occurred, and I reduced the number of species by 4. Okay. The Simpson's index or the Shannon-Wiener index is not half. Okay. It is not half of the Simpsons here, where I have double the number of species. The same thing here. If I have 16 originally, and there is, say, a storm, or if there is a disturbance, a pollution, and then half of my species died, and there are only 8 left, okay, then if I'm going to study the ecosystem, then I would say, oh, the disturbance was not really very bad, because the diversity, the Simpsons and the Shannon-Wiener indices tell me there was just a decrease in biodiversity of so much. Okay. So that is one of the disadvantages of the Simpsons and the Shannon-Wiener index. They are just numbers. Okay? They cannot tell too much about the structure of the community or the stability of the community, of the ecosystem. It even underestimates the effect of a disturbance in a community. Okay, so why is actually biodiversity important? Everybody's talking about biodiversity. So what? So it has to be high. The biodiversity is high. You have to maintain biodiversity. But why is it important to maintain a high biodiversity? So, well, they say that if the biodiversity is high, the resources are efficiently used, okay? Because of the diversity of functions of the different species. If there are more different species there, the resources in the community or in the ecosystem will be more efficiently used. Okay. And also it provides stability to the ecosystem. If the biodiversity is high, the ecosystem is more stable towards changing environments and disturbances. The ecosystem will have a high capacity to resist change, or it has the high capacity to recover in case there is a change, and there is a change to recover from a change due to a disturbance. So it is resilient, and the resistance is high. Okay. And biodiversity affects also ecosystem functions. What are ecosystem functions? Okay. So ecosystem function comprises all the stocks and fluxes of energy and materials in the system. Okay. So energy and materials, it's the resources in the system, and affects, therefore, the relative stability over time. So how does biodiversity do this? Everybody's talking about biodiversity. Okay, it provides stability, it provides high resistance and resilience, but how? What are the mechanisms? How does it do this? So I will try to show you some examples, but before going to that, there are most of, or a lot of ecologists now are doing studies on doing functional groupings. So what we do in school with our students, we do mostly taxonomic groupings of the organisms that we find on the lists. So that is the most common type of grouping that we use and introduce to our students, more on the taxonomy, more systematic. Okay? Non-functional groupings will be non-phylogenetic classifications of species. So it is be a classification of species not based on to what phylum they belong, to what family or what class or order what they belong. It's not non-phylogenetic, but it is a classification where the species which share the same characteristics and which share the same resources and play an equivalent role in the community will be classified together. Okay. The word here is equivalent, okay, because not if they are grouped together and their role in the community is equivalent. That means even if they are grouped in the same functional classification, each species still has different impact. Some may impact the ecosystem more, some lesser. Okay. And then another, so species, so functional groupings, they also, the species belonging to the same functional group will show more or less the similar responses to their environment or impact their environment. Again, it is not in the same way, but in a similar way. In a similar way because there are some organisms which are impacting more, okay, and some organisms which are impacting less. May it be due to size. Another, let's say for an example, let's put together all autotrophs together. Okay, we have the macroalgae, we have the microalgae, both are autotrophs. You have filamentous algae, macroalgae, and you have reef-like macroalgae. So depending on the number that they can be grouped together because they are all autotrophs. They are all photosynthetic organisms, and their function is production of oxygen, for instance, or producing biomass. So they are in the same group, but depending also on their number, for instance, or depending on their photosynthetic efficiency, they will impact their environment differently. So their contribution to the function of primary production will not all be the same. They will be contributing in the same way, okay, but their impact will be just similar to each other. Okay. So some of the biological and functional categories that we have that are being used by ecologists are this living habit, for instance, which means like if an organism is epibenthic, or is it attached, or is it an infauna. Mobility is about if the organisms swim, they crawl, they burrow, or are they sessile or sedentary. The body size, if they are small, medium, large, XL, XXL, so whatever, so whatever the t-shirt sizes like that. And then you have the flexibility. That means if it's soft-bodied or if it is rigid, it is hard or calcified. Feeding mode, if it is a predator, a sediment feeder, a filter feeder, a predator. Body form, if it is elongate, if it is round, is it encrusting, or is it like the algae, is it filamentous, is it leaf-like? Bioturbation, does it burrow into the ground, okay, or into the sediment, or into the substrate? And lifespan, does it live for years, for weeks, for months? So these are some of the categories which can be used for grouping organisms. And these categories, biological categories, are responsible for affecting or impacting some ecosystem function or process. For living habit, for instance, it can impact nutrient cycling, benthic-pelagic coupling, habitat structure, okay, or the sediment stability. So how stable is the sediment? Does it stabilize the sediment, or does it make it less stable so that it will be more prone to disturbance? Okay. And mobility, for instance, also will affect benthic-pelagic coupling, the structure of the habitat. Okay. Again, also hand in hand with habitat structure with sediment stability. The body size will impact biomass production, almost all of these functions. Flexibility will affect carbon sequestration and habitat structure. Feeding mode will affect nutrient cycling and benthic-pelagic coupling, and so on. Okay, body form can affect carbon sequestration, habitat structure, and sediment stability. Bioturbation, nutrient cycling, and sediment stability. So bioturbation, for instance, if you have nutrients already sedimented or already in the sediment, if they bioturbate, it can be possible that they are going to free some of the phosphates or the nitrates which have already settled in the sediment, or they can change the oxygen conditions in the sediment to help affect nutrient cycling. And lifespan, for instance, can affect biomass production, also carbon sequestration and habitat structure. Okay. So you don't have, so going back to this, all these biological functional categories, okay, will not have to be taken all in one study. So in a study or in a research, they just choose which of this is applicable to the type of question that they would like to answer in their research. For instance, here's one research performed by Val et al. About hard bottom dwellers, and they categorize the hard bottom dwellers with these four traits, according to the body size, the growth form, trophic type, and modularity. Okay, so one trait, the categories would be this. Okay, small, medium, large, XL, and XXL. And the growth form, it may be encrusting, massive, bushy, or filamentous, and the trophic type. So these I have said earlier, or modularity if they're solitary or colonial. And these are the functions in the environment or in the ecosystem which can be affected by this trait. So this is just an example of how to do this kind of research. So you don't have to name or to take all the categories or the traits that I have enumerated in the earlier slide. Okay, now here I will show you how biodiversity helps in stabilizing an ecosystem, or contributes to, high biodiversity contributes to stability of an ecosystem. An example would be through functional niche redundancy or functional niche complementarity, which is actually also not exclusive from redundancy. So there are some complementary relations which are also redundant. Okay. So in functional redundancy, there are several species. So I have here 1, 2, 3, 4, 5, 6 species which are performing the same function. So in this example, all these species are all helping together to move this block, that is the function, from left to right. Okay, so all of them are helping each other or doing the same function. So they are redundant, actually. But again, I have to point out that not all the species will be all contributing equally, but they have equivalent effects on this function. Okay. So there is a high degree of generalization in this type of relationships between the organisms, and this functional relationship among the species. Okay, and the species in this group are specialized to do the same function. If this is a resource, it may contribute to a higher interspecific competition, because if all of these species are using this resource, they will be competing for the same resource. Okay. So that is one of the drawbacks of functional redundancy. If there are more species using the same resource, then there will be more, or there will be higher interspecific competition. But if they are performing the same function, it will be to the advantage of the ecosystem, because more will be performing the same function. Why? Because we have them here. And if a disturbance occurs or something happens, okay, so if one of the species dies, okay, what happens to the function? There are still more species left to do the function. Okay, so at the start, probably it will be slower, but in time, a more resilient species will multiply faster, or will take the place of the species which was lost due to a disturbance, so that the function can go on. Okay, so in addition to these characteristics, in functional redundancy, the species are substitutable. Okay, so the blue one was lost, but it was substituted by the red species. and leads to ecosystem tolerance. Okay? Because even if there is a disturbance, only one or the less tolerant species will be eradicated or will be lost, but there will be others to replace them. And then it also leads to higher resilience, because after a disturbance, because the other species can take over the place of the lost species, the function would still be retained. Okay. So that in functional redundancy, at the start, okay, if there's still no functional redundancy, there is as you increase the species richness, the functional group richness also increases up to a point when there are enough species in the ecosystem that additional species will not change or will not affect the functional richness of the ecosystem. So a new species will not contribute to a new function, but will probably be grouped together with the other species already present doing a function. Another example of how biodiversity can, high biodiversity can cause stability of an ecosystem is this, through functional complementarity. Here we have two species, but these two species are specialized. One is a pusher and the other is a puller. Okay? They are specialized for doing this function. So their function is to move this block from down here, move it up here. Okay. So we have two species doing this function, but they are contributing differently to this function. Okay, so there is a high degree of specialization because they are specialized. They have specialized functions. The overall function is synergism. So whatever this one is doing and whatever this species is doing, the sum of their specializations, okay, the sum of their functions is to do this function. Okay. However, if the specialization is too high, this can also lead to the fragility of an ecosystem. Okay. So in this case, what happens if in the disturbance, one of the species dies? If the blue species is more sensitive to the disturbance. So there are two things which can happen. If species A, the red one, has a high tolerance for the disturbance, okay, it's possible that it can multiply, like in functional redundancy. The species can multiply faster so that the function can go on, so that the function would still be maintained. However, if species A is not tolerant to the disturbance, it may also perish, and there is no compensation possible. There is a loss of function and the loss of species. Another type of functional complementarity is a specialized non-conform functional complementarity. Species blue, okay, produces this product, this part of a puzzle, and species A produces specially just this part of a puzzle. But if you put these parts together, they form a function. Okay. So red cannot do the function of blue, and the same way blue cannot do the function of red. Okay. What happens if one of the species dies? The blue can multiply, producing all these parts, but the function cannot be completed because the part of the red species is necessary to complete this function. Okay, So here compensation is not possible. There is a loss of function. And depending on if this function benefits this species, there will be a loss of species because the function is lost. Also, again, if this function profits a third species, the loss of one of the specialized species can also lead to the loss of the third species. Okay. So in contrast to functional redundancy, in functional complementarity, more species increase the functional richness due to a high degree of specialization. Okay. So, well, it looks very complicated, but how am I going to apply this to my class? How can I apply this to the disk, which I am going to do with my classes, with my students? I have here an example of something that we did here in Kiel with some of our students. We placed discs in the water, and then we monitored them. So in March, April, and May, this is the first 2 weeks. In the first 2 weeks, 1 and 2 weeks, and then after 6 to 7 weeks, we took some plates again, and then after 10 and 11 weeks, we took the plates again, or the discs. So the number of species, we counted the number of species here from 2 to 9 to 11. Okay, so there is an increase in the number of species. So, what you will notice here is that we did not count the individuals. Okay, but we just counted the number of species. So, and then we classified the organisms that they found according to their feeding mode, according to their diet, and according to the mobility. For the feeding mode, this is the trait that we chose. Okay, so we classified the organisms if they are predators, grazers, shredders, sediment feeders, or filter feeders. For diet, are they detritivores, omnivores, carnivores, or herbivores? And for mobility, are the organisms sessile, semi-mobile, or mobile? So you don't have to make it very complicated, but you can just take one characteristic at a time. And you can see the development here. So you can see more about, you can see more information about the structure of the community or the ecosystem growing on the disc. In March, for instance, we just have the grazers in the first 2 weeks. So this is like a succession study also. So at the start, you just have grazers. Okay? Probably some of the ciliates, these are ciliates which are feeding on the diatom biofilm. Okay, so that's why we just have grazers here in the early stages of the fouling of the disc. And then in April, after 6 to 7 weeks, you have here a different kind of composition. You still have the grazers, okay? And then now we found shredders, and we have started to find filter feeders. And then after more weeks, the 10th and 11th week, the shredders are gone, okay? And the grazers are gone. Okay. If we say that the grazers and the shredders are actually mostly herbivores, okay? Or many of them are herbivores. And then here, the structure, again, of the community according to feeding mode has changed, wherein we have the filter feeders dominating the organisms, the species on the disc, followed by the sediment feeders. This might be the worms now. And then there are now also predators, because there are more organisms, bigger organisms now, which are can be eaten. Okay, I think, sorry. And then we have the diet. First, you have here the herbivores, of course, because these are the grazers, the herbivores feeding on the diatoms. So you can see again here that you have, the students can now see, so why are there grazers only? Because there are diatoms here, and we can see here that these grazers are herbivores. And these are mobile organisms, mostly ciliates, feeding on the diatoms. So you can see here the change in the community composition or the ecosystem composition from March to April. So they can make a better story out of this. So they can start to analyze and put the things that they learned in ecology from the classroom and see and try to explain why is there a change in the diet of the organisms, or the organisms which have different types of diets from the early part or the early stage of succession to the earlier or to the later stage of succession. Also take into account what part or what time of the year that is. This is early or early spring, and this will be already here in Kiel will be more or less early, starting to become warmer. And then also here, the mobility of the organism changes, where here we have an increase in the sessile organisms because we will already starting to have the barnacles. Okay, and then compared also at the different types of the year, we have autumn, winter, and spring. Okay, the first 2 weeks, blue is the number of species and red is the number of functional groups. For winter and spring, you can see that there are only 2 species. And again, we can already see that these 2 species, okay, are very different from each other. They do not interact too much with each other because they belong to 2 different functional groups. Whereas in autumn, you can see that here that there are 9 number of species, but there are only 4 functional groups. So that means that of these 9 species, there are some which share the same functional characteristics. So there may be already, here we already see functional redundancy. And then we classify the organisms again according to their feeding mode, the diet, and their mobility. And they can see changes already in the composition of species composition or the functional group composition of the organisms in the ecosystem. So this is just a simple way of how the students, or how you can apply these functional groupings in the school without necessarily having to count all the organisms or all the individuals in species, but you just have to count just what species are there, and then classifying them according to these functional groups, depending on what trait you would like to use: feeding mode, diet, and mobility. Feeding mode and mobility. So these 3 are the simplest ones which we can use in the school, because we have a lot of information where we can find this about the organisms, what they eat, how they eat, and how they move. So this may probably worth a try doing in the classroom. Okay, so moving on to the last slide. Okay, the penultimate slide. So what are the application of fouling discs in research? So we can say to our students that you are not doing that project just because so that they will be kept busy in school or they will have something to do in their science class, but it is for them really to learn how to work really scientifically, because it is not just them doing that, but it is also being actively used in research. Okay. So some of our scientists here at GIUMA also used disc, but in their research they use plates, PVC, polyvinyl carbonate, PVC plates, and they deploy them in the at the bottom of the Baltic Sea at different locations in the Baltic Sea. And well, you can probably motivate some of the students, but there's no diving probably in school. You can have other people do the diving for them. Okay, so some of the applications of fouling discs in research is like what we are doing here in GIUMA in Kiel. They are used for monitoring studies, or in some other areas, or in some other institutes, or there also have been studies also here in Kiel using disc for testing anti-fouling agents, or some may use the disc also to doing research on invasive species, or they can use the disc for comparing locations and also to determine the effects of changes in the environment due to pollution or climate change or ocean acidification and some other environmental changes. Okay, so to summarize everything, first, the disc, when they are deployed in a body of water, will first develop a biofilm. Okay, the biofilm will condition the substrate for later settling of other organisms, mostly macroorganisms. The bigger organisms may be soft-bodied or hard-bodied. So most of the organisms that will settle on the disc, of course, are benthic organisms, and they are sessile. A lot are filter feeders or sediment feeders. The microfoulers, these are the diatoms and bacteria, are followed by the macrofoulers, okay, which are soft-bodied or hard bodies, and these come from the water, okay, as larvae which are part of the zooplankton. The larval stages of these organisms that are found later, which you will find later on the disc, had a part of their life cycle as zooplankton in the water. So this zooplankton in the water will usually come from the organisms already present in that area, in that location. So if they start to give off their their eggs and sperms, and they will be fertilized in the water, they will develop into the water to form these larval stages, and they will become part of the zooplankton, and this will start settling on the disc if after the disc have been conditioned for settling with macrofoulers. Biofouling can cause economic costs as well as biological consequences, like the introduction of non-indigenous species. Okay? And usually the fouling in a certain area, the fouling of a disc or of a substrate in a certain area follows a predictable sequence, especially in the microfouling, the early stage, and that is called succession. And the virtue discs we have here in this project are very good and are suitable for calculating the most commonly used biodiversity indices like species richness, evenness, the Shannon-Wiener and the Simpson's biodiversity indices. But these indices, they are enough, okay? These are enough for school, but it should be noted that also these have shortcomings. A functional diversity analysis better describes the community stability and species interactions. Okay. And high biodiversity leads to a more productive and stable ecosystem, for example, through functional redundancy and functional complementarity. And different species have varying reactions to disturbances, and so it is important also to consider the identity of the species when analyzing community stability. And also then fouling on disc or plates are also used in research for monitoring studies or looking for fouling paint alternatives or biologic or some other chemicals which can be used for as anti-fouling, and also to study the environmental effects on the ecosystem. Okay, so that's it. So if you have any questions, you can put them, pull up your questions in the Canvas forum, and we can try to answer those. So I will be making a PDF file again of this presentation, so I can post them again. Tekla can post them again in Canvas later tonight or tomorrow. Okay.
Yes. Well, thanks, Sally. It was a pleasure to have you today. And thanks to all of you that joined us for this webinar. Soon we will post on Canvas platform the recording of this webinar. And you can also have a look at the updated version of the slideshow of Sally's talk. Please, as remembered also by Sally, do not hesitate to post any question in the forum of Module 2, where you can interact with Sally directly, but also with the other participants. And save the date for the next webinar, which is scheduled for the next Thursday, the 7th of May at 6 p.m. Central European Summer Time. I will be the presenter, and together with my colleague Juanita from Sumon, we will talk about ocean literacy and how Virtues this can be a useful tool to bring ocean literacy in your class. Thanks again for joining us today. Stay safe and take care, and see you next week. Bye. See you.
Bye.